My point is simple. When Dr. James Tour declares that origin of life researchers are clueless on origin of life, is he exaggerating? How do I explain to the non-scientist why this is such a huge challenge? This video comes in two parts. In the first part, I examine the simplest living organism. This organism was optimized in a lab to be as simple as modern technology can get. When we understand the target, an actual living organism, then we are ready to understand what hurdles naturalism must cross to explain it. The second part delves into the science of abiogenesis, the study of the origin of life. I don't get into chemistry and physics as much as I get into the differences between true and false and unconfirmed clues. I believe that you will find this model to be accessible to anyone with an interest in the topic, even if they have never studied science. So, on with the show. In 2016, the J. Craig Venter Institute announced that they had generated the world's simplest independently living organism, the JCVI Sin 3.0. This is approximately what they did. They started by reading the Microplasma genitalium, or M genitalium, DNA into a computer. Then they extracted the DNA from an M capricolum and replaced it with the computer's DNA. They placed the new organism in a pristine environment. It lived. It reproduced. It had the body of one organism and the DNA of another. They accomplished this in 2010 and called the organism the JCVI Sin 1.0. This was a major breakthrough, as now they were able to make any change they wanted to the computer's copy of the DNA and see what happened. Once they had this technology, they embarked on a venture to make the simplest independently living organism. They extracted one gene from the computer's DNA image and put the result into the DNA-less M capricolum. If the new version lived, they concluded that the gene was unnecessary, at least for basic life. Then they tried the next gene. If the resultant organism did not live, they decided that the gene was necessary, so they undeleted the gene. They kept doing this, effectively playing whack-a-mole with the genes in the computer's DNA image. In 2016, they were done. They called their organism the JCVI Sin 3.0. It had 531,560 base pairs, 473 genes. 149 of those genes had unknown function. I decided to visualize this a little bit. I downloaded the computer version of the DNA. I then printed it as colorful bars, red, green for AT, blue, yellow for GC. By flipping the bars upside down, I was able to render a nice approximation of the DNA. I initially envisioned 3D printed Lego-like blocks, but the task proved ominous. I tried printing where each bar was 1/8 inch wide. Still, the results were enormous. It would print out at over 2,000 sheets. I settled on 1/20th of an inch wide bars. That was the smallest I could make and show the letters readably. I also created a 3-barred code to represent alphanumerics. This is similar to the 3-base pair amino acid system that DNA uses. I paralleled the DNA with the novel The Call of the Wild by Jack London. The results are printing along the bottom of the screen. If you pause the video at any point, you can see one page worth of JCVI Sin 3.0 and one page worth of the novel. So let's see how dense this code is. An American quarter covers 25 base pairs. A quarter is a bit thicker than a printed bar, So a stack of quarters representing this DNA would cost over $130,000 and be over 1,100 feet long. Let me address a major misconception that I used to have. I used to think that genes were tiny things. If we take 531,000 base pairs and divide by 473 genes, we get well over 1,000 base pairs per gene, or over 333 amino acids per protein. There is some required epigenetics in the organism. Typically, a gene is 150 aminos long, with 450 base pairs. So it appears that the genes in the JCVI SYN 3.0 are particularly large. So let's look at the volume of information in one gene and parallel it to the text. You can see that it parallels to about a paragraph of text. It contains the careful instructions to make one 3D part. Each 3D part is radically different from other 3D parts. Very often these parts work in conjunction with other parts to do a particular task. Now notice also that Venter says that about 1/3 of the genes have unknown function. However, without these genes, the cell dies. There is a lot more research to do just to understand this simplest organism. The DNA printed out on 1,068 landscape pages. Each page renders 10.5 inches of DNA. I got friends and family to help me tape these sheets together into a mighty bifold. We then found a quiet street and spread out the printout of the DNA. We placed rocks on every second or third page so that they wouldn't blow away. You will notice a fast motion video of the rollout in the upper left corner of the screen. By the time we got this monstrosity rolled out, light was seriously waning. In any case, you can see that the printout goes on and on into the black. Yes, the DNA required to make the simplest life form on Earth is, well, enormous. Some conclusions. This organism is the closest we have come to the legendary Luca, the last universal common ancestor. There's something we need to understand about Luca. Bacteria and archaea separated about 3.5 billion years ago. They share most of the same genes. Therefore, this technology was fully developed 3.5 billion years ago. So this is not the product of billions of years of evolution, maybe 100 or 200 million, but not billions. So all life that we know, whether in nature or generated by man, is just amazingly complex. This has been scientifically tested to be irreducibly complex at the gene level. Therefore, most, if not all, of the multi-gene subsystems in this organism are also irreducibly complex at the gene level. In each of our living cells, we have 3 billion base pairs from each parent. This encoding scheme renders 1,267,200 base pairs per mile. An entire listing of one parent's contribution to our DNA would print out 2,367 miles long. So if we printed it out, it would stretch from Anchorage, Alaska, all the way to Disneyland. Whoa. As I look at my next video, I want to address where science is really at regarding origin of life. Can scientists generate life in the lab? Dr. James Tour claims that origin of life scientists are clueless about how life originated. Is he right? With all of the headlines claiming again and again that finally life's origin has been solved, how can we really get a true answer to this question? I promise you, we will. Even if you are a non scientist, you will be able to be confident of where origin of life research is at now and into the future. Here's the deal. If you follow science news as I do, you will have encountered hundreds of articles declaring that some aspect of origin of life has been solved or may have been solved. If it weren't for the fact that the next article that comes along declares the same victory that the previous article declared, you may conclude that origin of life, abiogenesis, is settled science. Then you hear this voice crying in the wilderness declaring that origin of life scientists are clueless on origin of life. Clueless? Really? With all those articles declaring that clues have been found? And when there are two divergent declarations, how am I, as a non-scientist, supposed to determine which declaration is correct? Must I learn all there is to know about OOL research? Should I ask my high school biology teacher? Would he know? Let's listen to Dr. Robert Hazen, an origin of life researcher.
That if you decide you're going to go into this field, you better believe that this is true. And that's belief. This is not truth. It's just a philosophical stance.
Wow. He says you must believe, i.e., you need a faith position. Clearly, he doesn't think that this is a solved problem at all. Let's look into the literature. This was published in the journal Nature Review Chemistry. I know, 2017. Ages ago. Not really. Remember, the Miller-Urey experiment, the serious beginning of OOL research, was in 1957. So this article shows where OOL is at after 60 years of research. Look way down in the bottom here, the current state of the field. Whoa. And it goes on to say that we need a major system innovation. That looks awfully like we're clueless. Now we see an area called chemical necessity. So this need for a major system innovation is well inside the pre-life territory. This is the area where Dr. Hazen has been working. It is possible that he has moved up the line a bit. As we follow the line up, we see fully alive. Okay, but we know that the JCVI SYN 3.0 is the simplest living organism known to man. So we know that there is no actual knowledge of where this line fully alive belongs. Could it be that the break lines belong below the fully alive point rather than above it? But are we really that clueless? Oh, heavens no, there are hypotheses all up and down this line. So we have lots of clues. What on earth, then, is Dr. Tour talking about? He says, look, nature needs to be able to produce these 5 phenomena. Useful polypeptide synthesis, useful nucleotide synthesis, useful polysaccharide synthesis, specified information, assembly of a cell. He placed this challenge out to the abiogenesis research community. His offer was most generous. If 3 top researchers agreed that any one of these challenges had been met, even in a painfully artificial environment where we are given all 19 canonical chiral amino acids, the nucleotides, the monosaccharides, all with 100% purity. This given is extremely generous. This is the kind of stuff that the Miller-Urey experiment makes, but by no means with 100% purity. Far, far from it. Dr. Tour placed this challenge out to the OOL research community in August 24, 2023. His position: if a jury of 3 OOL researchers agreed that any of these challenges were met, he would remove all of his OOL is clueless content. As he has not removed this content, we can be assured that OOL scientists see all 5 of these to be unmet challenges. But here's the problem. Each of these are clues sitting on our chart. Most of these have published literature saying, we've got this bit, but obviously they don't. Each of these clues are potentially false clues. Do you remember our von Neumann life discovering a rock that was zapped with lightning which contained a strand of copper? Yes, he found a false clue. False clues are a real problem. If you think about it, some of the clues on the chart must be false clues. I mean, some researchers are saying that life began near hydrothermal vents. Others are saying that it started in the intertidal zone. Still others say it started between sheets of mica. At most, one of these hypotheses could be right. Therefore, at least 2 of these must be false clues. I remember back in the 80s when the RNA world hypothesis was all abuzz. This was so cool. Now the information was the duplicating machine itself. When we look at the 5 essential components of life, we have merged 2 of them, the computer and the information. I remember reading all manner of articles espousing wild things happening in RNA world labs and wondering why they kept calling it an hypothesis rather than a theory. It was an exciting time for OOL. But let me show you what Dr. Hazen has to say about the RNA world.
Well, the RNA world dilemma is that, it's a great way to potentially make copies of yourself. It's a great way to carry information. It's possibly a great way to catalyze reactions. But there's no known way to synthesize it. No one has been able to figure out a way how to do that. And so you really have to figure out, you've got the prebiotic soup with all these potential building blocks of life, and then you've got the RNA world, and that gap's a big one to fill.
He is motivated to believe in the RNA world because it doesn't challenge his research. So when he says that the RNA world hypothesis remains implausible after 30 years of experimentation with it, I've got to conclude that the RNA world is potentially yet another false clue. So here we sit. We've got clues all over the place. We know for sure that some of them are false clues. But we don't know that any of them are true clues. None of them. Well, we know that certain phenomena could happen in the prebiotic world, things that appear interesting. But we don't have any good scientific reason to believe that these phenomena were direct precursors to life. We only have faith. Because without faith, it is impossible to do OOL research. If we have no confirmed true clues, is Dr. Tour exaggerating when he declares that science is clueless on origin of life? I, for one, am with Dr. Tour on this one. So you say, but you promised to leave us non-scientists certain that we are getting the straight scoop from you. Ah, yes. How can you know that you've got the straight scoop? Watch what happens when we type origin of life or abiogenesis into Wikipedia. Look at the provided illustration. Remember, a picture is worth 1,000 words. What are these great big question marks doing in there? They are declaring that science is, well, clueless about origin of life. Let's try another tool designed to clarify science to non-scientists. Our friendly neighborhood AI bot. I asked Microsoft Copilot, has man made life in the lab? It responded, yes, scientists have made significant strides in creating synthetic life forms in the lab. Here are some key developments. 1. JCVI SYN3A. 2. Artificial DNA. Effectively, they added a man-made protein or two into the JCVI SYN. Please note that with JCVI SYN3A, they state, replaced it with DNA that was designed on a computer and synthesized in a lab. Well, you know that's an exaggeration, as Craig Venter's own website declares that 147 of the genes are necessary but have unknown function. They certainly can't have synthesized those in a lab. Clearly, while the DNA is a unique subset, the individual genes in the JCVI SYN 3.0 were all written by nature, not by man. So we can see that these sources are exaggerating. They are motivated to declare success. If they are saying that the issue of life's origin is not solved, we can be confident that it is not solved. So I asked Copilot, is origin of life understood by science? It responded, no. There are still many unknowns. How life began. The process by which life began is much less understood. There are several hypotheses. It then lists some hypotheses that are incompatible with each other. By point 5 it says, artificial life. Science have made significant strides in creating synthetic life forms in the lab, which can provide insight into how life might have originated. Again, they are referencing the JCVI SYN3 as the exemplar of where origin of life research is at. So please understand, these resources, Wikipedia and Copilot, are designed to communicate to the non-scientist. They are motivated to give you the most positive possible impression of where OOL is at. As long as they are saying that this is an unsolved problem, you can rest assured that it is. Clueless? As long as they are using the word hypothesis rather than theory, you can also rest assured that there is no confidence that the hypothesis is a true clue. As long as the only things available are questionable clues, I think it reasonable enough to declare that OOL scientists are clueless on origin of life. So am I suggesting that the only logical conclusion on this matter is that science is clueless on origin of life? I'm really not. However, if you understand that all known living organisms are just enormously complex, and if you understand that while science has many hypotheses about how it developed, it is with good reason that none are called theories, then your position is scientifically valid. If that doesn't translate for you as clueless, so be it. But please don't pretend that science has origin of life solved, or even nearly so. In my next videos, I will reveal all of object-oriented programming in DNA. I will show you examples where DNA implements procedures, if statements, while loops, classes and instances, encapsulation, polymorphism, inheritance, databases, and even abstraction, generalized algorithms. Some of these features, I am convinced, are way beyond what is possible within the Darwinian paradigm. I am convinced that what I am seeing proves that the Darwinian model is fundamentally inadequate as an explanation. The Canadian subarctic is beginning to thaw, which will draw my attention in other directions, so I'm not sure how quickly I will be able to get to this material, but it is very much my intent. Please like, subscribe, comment, and share this video.